let's talk about the nervous system. So first off, what is the purpose of the nervous system? Well, as humans, we must be able to perceive our surroundings and then act on that information. For example, when you place your hand on a hot stove, sensory neurons in your hand are going to detect that heat and send the information as an electrical and chemical signal to your brain. Your brain will then be able to send that information through motor neurons to stimulate you to move your hand so that you don't burn it. In this way, the nervous system is critical to trigger our involuntary and voluntary responses. But what are the building blocks of the nervous system? The nervous system is going to consist of neurons that are all sizes and shapes. Let's take a look at the general structure. So from left to right, we have a neuron that has these branched extensions called dendrites. And these dendrites are going to receive stimuli from various sources in the body and even from other neurons. So a neuron also has a cell body where the nucleus is found. This right here is the cell body. And as we move further down, we will come across the axon hillock. So signals that come from different dendrites are going to accumulate or sum up at the axon hillock. And if the strength of the stimuli is great enough, then an action potential is fired and it will travel down this long extension called an axon. Now, axons tend to be really long so that the signal can travel far distances. And along the axon, we have bubbles of insulation called myelin sheaths. So these myelin sheaths are going to prevent the electrical signal from being lost. And in that way, they're insulating the axon. Between each myelin sheath are gaps called nodes of Ranvier. And these nodes of Ranvier are going to have special channels that are needed for the action potential to occur. The action potential is able to jump over the myelin sheaths and in between the nodes of Ranvier. So, this structure allows the action potential to propagate faster down the axon and in only one direction. The signal will eventually reach the axon terminal, and the axon terminal is the end of this neuronal cell. So this right here is the axon terminal, and then the space between the axon terminal of the first cell and then the dendrites of the second cell are called the synaptic cleft. And here we have neurotransmitters that are chemical signals that will be released so that the signal can be transferred to the next neuronal cell. And so overall, we see that the action potential travels in the direction of the dendrites and the cell body through the axon to the axon terminal. And also, the neuron transmitted an electrical impulse and converted it to a chemical signal in the form of neurotransmitters. But keep in mind that neurons can follow this structure to perform their functions, but they do have different shapes and sizes throughout our body. And this is what enables our nervous system to be so complex. But what does a neural cell look like before it receives stimuli and after it receives stimuli? Well, they have something called a resting membrane potential. A membrane potential is the difference in electrical charge across the membrane. So, in simple terms, if we placed an electrode on the inside of the cell next to the membrane and another on the outside of the cell, we would be able to measure the cell's electrical difference. And remember that cells contain different charged ions. They can be cations or anions. And so the cells are going to have a certain voltage. So the resting membrane potential is the cell's net electrical potential difference across the membrane. And for a neuron, the resting membrane potential is negative 70 millivolts. But what ions and channels contribute to this resting membrane potential? Well, two most important ions are sodium and potassium. The sodium and potassium pump is going to actively move 3 Na plus out of the cell, and at the same time it's going to move 2 potassium or K plus into the cell. So the sodium and potassium pump is actively moving these ions into and out of the cell against their electrochemical gradient. And it's important to remember that they are moving against their electrochemical gradient, because this is creating more negativity inside the cell, along with the already negative proteins and nucleic acids that are not pictured here. Now, potassium ions are also going to diffuse out of the cell through leak channels on the membrane. And since we're moving positive ions out of the cell, we are leaving more negative charge inside the membrane. And finally, there are these sodium leak channels along the membrane, but way less of them than the number of potassium leak channels. And sodium leak channels are going to let Na plus enter the cell. But since there's way less Na plus entering the cell, we are removing more cations, as in K plus, than we are bringing in. So the key channels that maintain resting membrane potential are the leak channels and the pump. Now, since we're talking about membrane potential, let's talk about what happens when membrane potential changes. So if the membrane potential becomes more positive than resting, it is called depolarization. So if we went from negative 70 to negative 60, let's say, we would be depolarizing because we're becoming more positive than resting membrane potential. And if our membrane potential becomes more negative than resting, the cell is hyperpolarizing. So if it went from negative 70 to negative 80, that would be hyperpolarization. So we've looked at the structure of a neuron and also what the membrane potential of that neuron looks like when it's at rest. So let's now consider how the membrane potential changes when we do have an action potential. But what is an action potential? So an action potential is what happens when we want to send a signal down a neuron's axon so that we can pass the signal to other parts of the body. Now currently the cell is at resting membrane potential, which is negative 70 millivolts. And in a neuron cell, we have 5 main channels on the membrane: the voltage-gated sodium channel, sodium leak channel, potassium leak channel, and sodium-potassium pump, and then voltage-gated potassium channel. So the pump is moving sodium out of the cell and potassium into the cell against their concentration gradients. We also have potassium leak channels moving K plus out of the cell down its gradient from high to low. And then we have a few sodium leak channels that are moving sodium into the cell, but very little. However, voltage-gated sodium channels and voltage-gated potassium channels are closed. They are not moving any ions into or out of the cell, and we have negative proteins and nucleic acids inside the cell, so overall our resting membrane potential is negative. Let's now look at what happens in the depolarization phase. So this dotted line shows the threshold potential, and threshold potential is negative 55 millivolts for a neuron. So in order to send an action potential, we must reach this threshold potential. Let's say that we get a stimulus that causes our resting membrane potential to change from -70 to -55. We have reached threshold, and we are now going to depolarize the cell. So remember that sodium and potassium leak channels were open at resting membrane potential, and the pump was also open. So all three are open, and they're going to stay open. But the main thing that happens is that now voltage-gated sodium channels are going to open. And voltage-gated means that they only open when there's a change in the membrane potential. So when these channels open, sodium ions are going to rush into the cell. And since you're bringing so much positive charge into the cell, the electrical charge inside the cell is becoming more positive. So this is called depolarization. Remember we said depolarization is when the membrane potential becomes more positive than resting. And here it's becoming more positive than -70, and it's actually reaching +30 millivolts. The key thing to remember is that this positive charge inside the cell comes from the sodium ions rushing down their gradient into the cell through voltage-gated sodium channels. Now, what's going to happen when we reach this peak on the action potential? When we reach this peak, we are going to begin repolarization. Remember, sodium leak channels and potassium leak channels and the pump are always open. They're going to stay open like normal. But the key change is that when we reach the peak, voltage-gated sodium channels close and voltage-gated potassium channels open. Remember that we had so much K+ inside the cell and very little outside the cell. So as soon as these voltage-gated potassium channels open, K+ is going to rush out of the cell. And since you're removing positive charge from the cell, the inside becomes more negative. And therefore, on this graph, we see that we're going in the negative direction. This is the resting membrane potential, and repolarization is moving back towards resting membrane potential. Now, after repolarization, we reach hyperpolarization. And here we let too much potassium out of the voltage-gated K+ channels. So the inside of the cell became too negative because we let too much positive charge out. And these voltage-gated K+ channels will close. So this stage is called hyperpolarization because we are more negative than resting membrane potential. Remember, resting membrane potential is -70 millivolts, but we are below that. And this is also called the refractory period or overshoot, because at this point, no other action potential can be sent, and the action potential is also prevented from going backwards. Now, eventually, the pump and leak channels that were open the entire time will help us return to resting membrane potential, and the pump is key for this. Remember that the voltage-gated channels are closed. One of the most important ideas about this graph is that an action potential is all or nothing. So that means that once we reach threshold, the shape of the graph will look like this. So the action potential of this exact size and shape will be sent. We can't get a peak that is less than +30 millivolts or a peak that is more positive than +30 millivolts. So this is where the peak will be, and this is what an action potential shape will look like. So finally, we've sent this signal as an electrical signal along the neuron, but how do we send it to the next neuron so that the signal can travel to the rest of the body or to our brain? Well, we must convert this electrical signal to a chemical signal, and this is going to occur at the axon terminal and the synaptic cleft. So here's our neuron and the axon terminal, and here's a zoomed in version of the axon terminal and the synaptic cleft. So in this zoomed in picture, we see that when the axon is depolarized, an action potential will be sent, and that action potential will arrive at the axon terminal. This depolarization is going to cause voltage-gated calcium channels to open at the axon terminal, and calcium is going to rush into the cell. So this increase of calcium concentration inside the axon terminal is going to cause vesicles of neurotransmitters to fuse with the presynaptic membrane. And neurotransmitters, which are chemical signals, are going to be released into the synaptic cleft. Neurotransmitters are going to travel along the synapse, and they are then going to bind receptors on the postsynaptic cell's dendrites. So here we see the receptors that are ligand-gated ion channels, and that means that they require a molecule to bind to them in order to open. Now, when these channels open, they're going to allow sodium to enter the next cell. So here we have sodium entering the cell, the postsynaptic cell, and we also have sodium here, and this is going to depolarize the next cell and allow the cell to fire an action potential that continues to send the signal. And that is it. That is how our neurons sense something from the environment and send the information to our brain, or how the brain sends information to our muscle cells. Our cells do this in milliseconds. I really hope that you're able to gain an appreciation for the nervous system through this video. Thank you for watching. Please subscribe, like, and comment. Thank you.